excited to introduce you to our Omics Logic Training programs in Nigeria. This program will help you see the role of big data and next generation sequencing technologies in life sciences. Omics Logic Bioinformatics Training is designed for everyone interested to learn and apply data science skills to study biological datasets in an effective and meaningful way. Before we get started, let me introduce you to our company vision and team. My name is Beepsha Biswas, and I am the Omics Logic Community Manager responsible for daily interaction with thousands of users that are part of a bioinformatics community managed by Pine Biotech. Let me start by introducing Pine Biotech, the company behind Omics Logic, share with you details about the upcoming Omics Logic programs, and explain how the training resources we will discuss today will be used in the upcoming training program. Omics Logic is a program for bioinformatics training that was developed in collaboration between Pine Biotech and the Tauber Bioinformatics Research Center. What we will discuss today and throughout this program is a result of hard work by an amazing team led by company co-founders Dr. Alfred Tauber and Dr. Leonid Brodsky. Dr. Tauber comes from a medical background and has extensive expertise in oncology, immunology, and biochemistry of inflammation, and has been recognized internationally for his generous support of many research projects and initiatives that support data science training and applications in medicine. Dr. Brodsky is the director of the Tauber Bioinformatics Research Center at University of Haifa and is an expert in bioinformatics and biostatistics. He has numerous publications in mathematics and bioinformatics, and over the course of his career has developed multiple novel algorithmic approaches to biomedical data analysis. The rest of the team you see here includes experts in bioinformatics, computer science, and training, and some of our team will be presenting today as well. Over the course of the past several years, our team has been collaborating with a number of academic institutions around the world that have participated in content evaluation, curriculum review, and project design. As a result, the Omics Logic programs have been thoroughly evaluated and are known to lead to excellent outcomes. Importantly, the programs are designed to support students with minimal bioinformatics background, including biology and biomedical studies majors, as well as faculty interested in bioinformatics, data science, and biomedical research. The goal of the Omics Logic programs is to provide training in bioinformatics, enabling independent research guided by mentors and peer example, developing a growing community with shared passion for data-driven research and appreciation of citizen science, enabling students, clinicians, and faculty of all backgrounds to develop novel and independent research using latest technology in life and data sciences. In this webinar, our team will introduce you to resources that will get you started on this journey. We hope that you will be able to benefit from this webinar. The objective is to get you started. So let us go first to this URL that you see on your screen, edu.t-bio.info. Dr. Mohit, if you can kindly post the link on the chat. So we would like for every one of you to go to this link and sign up. If you're already signed in and you already have an account, then please log into your account and be logged into your account through the rest of this process, because we will be showing you how to go about with the different resources during the course of this session today. And if you have been able to sign up, please put 1 in the chat box so that I can understand that you have actually signed up to this portal or you're already logged in. Thank you, Dr. Mohit and Aditayo, for sharing the link. Has anybody been able to sign up or log in to the portal? Okay. So moving on, as you can see, you will be able to log in using your username and password, or also connect using any of your social media profiles. I see already Francis and Somaya has done this. So as we progress with our session today, please make sure that you're already signed into this site. It is a free registration. Now moving on, we are excited that this event has been led and organized by a dedicated team in Nigeria that brings together experts who have an interest in data-driven research and have all recently completed our specialized training. They are now certified and ready to assist all of you in joining the program, getting registered, finding all the necessary resources, and helping you troubleshoot any technical or conceptual challenges you come across. The program will also be supported by a team in the US and India. Dr. Harpreet Kaur is a certified Omics Logic trainer and expert bioinformatician who will guide the hands-on sessions. Dr. Mohit Mazumdar is our Omics Logic project mentor. Ilya Brodsky is Pine Biotech co-founder and CEO. I will now pass it on to Ilya to talk about the increasing significance of data science in biomedical research and introduce you to the types of challenges we will be addressing in this upcoming program.
Thank you very much, Bipsa. And let me share my screen. And welcome everyone to this webinar. It's exciting to see that so many of you were able to join. And Bipsa, as I'm going through some of these slides, please be ready with the questionnaire so that we can get to know some of our participants. So today we are here to discuss bioinformatics. And the natural question is, what is bioinformatics? And the truth is that bioinformatics is this intersection between informatics technologies or data science, statistical analysis and analytical approaches, and a focus on biological data. And biological data is unique data. It's not just data like marketing data or finance data. There are very special types of data that we have to know how to process, prepare, and manage effectively when it comes to bioinformatics. So the bioinformatics approach is the intersection of all three. And a lot of people confuse bioinformatics with computer science or with biotechnology, but it's actually right there in between. So let's take a look at some of these data sets and try to think about how has the nature of data in biology changed since the development of next generation sequencing technologies or high throughput technologies. Since the project for the human genome, there has been an exponential growth in the types of data and the types of applications of big data in biotechnology, in biological research, biomedical research, in clinical applications. So there has been this challenge of big data, which is heterogeneous or diverse types of data, which are growing in the number of samples and the size per sample. And also it is a continuous inclusion of new types of variations. So we go from cell lines to animal models to patients, now patients around the world, sequenced around different continents, different ethnic backgrounds, different diets, different conditions, et cetera, et cetera. So it's really an exponential growth of complexity. And a good way to look at this is to see how this has changed since this new pandemic. So in this new pandemic, we have seen a tremendous growth of publicly available genomes for the SARS-CoV-2 virus as compared to some of the previous pandemics like SARS, Ebola virus, MERS, and Zika. And what we see today is that even the general public is aware of the genomic properties like new mutations. They understand the different proteins. They understand how they interact with the host. And here we are coming from a biological or biomedical background and have now access to all of these amazing data sets that we can leverage to conduct our own research. So what types of biological phenomena could be studied with omics data? When we think about omics data, we are really talking about a number of different types of omics data. For example, we can think about phenotypic data or phenomics. Phenotypic data describes the external characteristics of cells, tissues, and organisms. So we can use clinical data to collect phenomic data. We can use imaging. We can use any kind of an external characterization of whatever tissue and samples we're studying. And then we have what's called genomic data. Genomic data characterizes the DNA code. And DNA code could have a number of different variations that include single nucleotide variations or single nucleotide polymorphisms. It includes copy number variation, different types of repeats and segments that are studied separately. Then we can also look at epigenomic data. Epigenomic data includes data that characterizes different epigenetic regulation mechanisms. For example, we can think about whole genome methylation patterns. So how is DNA methylated, or how is the chromatin condensed or expanded so that you can actually express some of those accessible regions? And that is being studied through immunoprecipitation. So many different technologies. And ultimately, we can also think about the transcriptomic data, or data that describes gene expression. And that gene expression could be different levels of gene expression, different types of transcripts as a result of alternative splicing. So you can see that just using next generation sequencing, we have access to a whole range of data types that we can study. And the key factor about all of these types of data is that all of these types of data have to be processed. So when we talk about whole exome or whole genome sequencing, when we talk about DNA methylation and ChIP-seq, RNA-seq, metagenomics, proteomics, metabolomics, one key factor in all of these data sets is they have to be prepared for analysis. They have to be processed. So we have to understand what is the data that is being collected, how to process it so that we can separate signal from noise, and then what are some of the downstream analysis options and biological interpretation. So this program that we're introducing in this webinar is going to focus on addressing all of these different topics by giving you a broad introduction to a variety of different types of omics data and giving you resources and tools so that you can learn more in depth about each one of these and understand how to apply them in the context of a practical project. So the program that we're introducing today will consist of three important elements. First one, we will have interactive sessions just like we have today. We will meet on Zoom. We will discuss some of these omics data types, and we will look at how to practically analyze such data. The second part of the program are the self-study materials that you can find on that portal that Bipsa just mentioned and you have signed up. You will find there such resources as Introduction to Bioinformatics, Transcriptomics training that includes 4 different courses, Genomics training, Metagenomics training, and many others. Importantly, these are not just theoretical materials, but they're also facilitated by practical exercises on our T-Bioinfo platform. The platform is guided by artificial intelligence to enable you to conduct big data analysis research in the context of very specific types of data sets. But it doesn't end there. We also will include practical assignments in coding. So those of you that are more interested in the data science and the application of data science to these types of biological data sets, we will give you access to some practical coding exercises so that you can also leverage such practices as coding in R and Python to investigate some of these data sets. So let's take a look at how you can learn about some of these omics data sets and give you some examples that you can kind of experience what will happen during the program itself. So the first thing that we'll do in the program is we'll give you an introduction of some of these computational tools and data sets that you can leverage. To do that, let me go to the Bioinfo platform that we will talk about throughout this course and throughout this webinar. So if you can go together with me to server.t-bio.info. Please let me know once you are there. You will find here that you have access to many different types of omics processing pipelines, including mass spec data, structural biology data, and downstream analysis sections. For example, we'll take a look today at an example pipeline for processing RNA-seq or transcriptomic data. Once you get to this example, we're going to just do a demo, so don't worry about uploading any files. And I know that many of you don't have access to this, but you will have access once you join the program. And if you would like to have a demo account, you can go ahead and request a demo account right here using this access form. So once you start the process of analysis, you need to prepare the reads or the raw data that is coming out of the sequencer. Once you have prepared, you will find a couple of options that are available for pre-processed reads. Then we can quantify by mapping on transcripts and preparing a table of expression. And once that table of expression is prepared, you will find the results in the output section right here. So now we are ready to do downstream analysis. Now, this platform is a big part of what we are trying to do, because for anyone without any technical background, you can get access to these advanced computational tools to study a variety of different types of data sets. So again, as Bipsa mentioned, an important part of what we are trying to do on this webinar is to get you started. We want you to sign up on these different resources that we are going to be discussing throughout this session, so that when the program starts, you're all ready to get going. So hopefully by now you have signed up on the educational portal. I just showed you the practical analysis pipeline where you can request access, or if you have already joined the program registration and have completed it, that account will be sent to you. And in a few slides, I will introduce you to another resource where you can learn about coding. But let's talk about how can these different resources help you study, for example, genomic data. The human genome is over 3 billion base pairs long, with over 20,000 genes and many unexplored dark matter sections, junk DNA, that is still being studied. Learning about the impact of variants like single point polymorphisms, copy number variation, and even repeats is still a challenge that many teams around the world are studying and exploring. So knowing that each sample is a huge data set that has to be separated from commonly found variations and unique variations that could be associated between the variants and the phenotypes. And so to help you understand what goes into preparing such data, sequencing it, and then measuring some accumulation of mutations in conditions like cancer or phenotype or haplotype based on population, you can find a number of different pipelines that we will discuss and that are in detail explained in the Introduction to Genomics, Genomics 1, and Genomics 2 courses. We'll also cover an important section of transcriptomics that I just showed you an example of, where you will find processing pipelines, data preparation techniques, analysis and exploration of variation between different samples, and such common approaches for biological hypothesis testing like differential gene expression and pathway and gene set analysis and enrichment. We'll also talk about metagenomics, where we understand the type of microbiome by studying the sequences of 16S ribosomal RNA to understand the microbiome by looking at the diversity and composition between different groups of samples. So we'll actually take a look at some healthy microbiome for example, in a citizen science project called American Gut, and also think about how can microbiome play a role in multiple different conditions. These conditions can be subtle, like actively exercising or inactively exercising, maybe sensitivity to different types of foods, or even such diseases like cancer, when we talk about treatment, or Alzheimer's disease, when we talk about the condition of the brain and the brain microbiome, or how the microbiome in the gut could be associated with the health of our mental and psychological condition. So all of these exciting tutorials and projects are described in the educational portal, and then you will have access to interactive sessions where our mentors will be describing these projects in detail and helping guide you through the process of analysis. Now, you've probably heard about another paradigm shift in the area of big data, and that has to do with machine learning, analytics, and data science. And so we understand that the challenge for many people to start getting used to this concept of data-driven research is that a lot of times we have access to data without a predefined hypothesis. But what we do have is access to a data set, tools that can help us explore patterns, and then convert those patterns into a specific training for a computer to learn from. And so we've introduced a number of data science resources and coding challenges that will help you get started with R and Python. To kind of show you what that will look like, let's go to another resource that is going to be used throughout the session. So we've looked at the educational portal, we have looked at the server big data platform, and now let's go to code.omicslogic.com. And this is also a website that we use throughout the upcoming program. And you can log in and create an account here for free. So once you get here, you will see a place to sign up. And once you're signed up, you will gain access to courses. So throughout this program, we'll touch on many of the code examples that you can then modify to make sure that once you've explored the data, you can also present your analysis results in the context of what you are trying to do with effective visualization and analytical tools. As an example, we have here a couple of courses that are for introduction to data science in R and in Python. And all of this information is linked to the different types of examples that are being covered in the educational materials that we will review throughout this program. It starts from very basic stuff. You just load files, explore the files, understand different data types, and goes all the way to advanced machine learning methods that you will gain access to as you register for the program. And Vipsa will explain at the end of this session how to complete your registration so that you get access to both free and premium resources in the context of this full program. So another resource that I encourage all of you to already sign up for and try it out. So how will all of this work in the context of this program? In the context of this program, we will cover 6 different sessions, and you will have access for the period of this whole program to all of these different resources and get guidance on how to use them effectively in these sessions. We'll start by introducing next generation sequencing, a major data type that has completely transformed molecular biology, biotechnology, agriculture, neuroscience, oncology, and many other domains. We'll talk about how bioinformatics and big data is playing a role in biomedical research and practice. We'll then talk about the synchronous courses that are around collections of data for genomics, transcriptomics, metagenomics, epigenomics, and others. We'll cover some of these practical steps of analysis in detail. And then we'll introduce you to important project examples that you can try on your own, developing your practical skills and seeing how they're applied by experts to address specific challenges in big data sets. Importantly, these resources are all linked together. So what you see here is the overall process that we have tried and refined over the course of the past couple of years with thousands of participants from around the world. All of these types of programs are structured in the following way. Your activity from all of these platforms, from the server platform, from the educational portal, from the coding challenges, is aggregated into your individual profile. And as a group, we go through sessions where, as you can see, the yellow here represents the educational materials, the gray represents the practical analytical pipelines, and the pink right here represents coding exercises. So you'll see how all of these materials together will help you go from an observer that understands the different challenges, projects, and data types to then practically apply them in the context of predefined examples, and then get introduced to independent projects that you can develop into a research study of your own. For example, just recently, we have done a number of programs around Africa where we had multiple participants taking a look at some of the courses like what you're looking at today, Introduction to Bioinformatics, Transcriptomics, and others, and then look at such project examples as the Ebola project and the origins and pathogenesis of the novel coronavirus. So a project that helps us explore variation at the genomic level of these pathogens and try to find association with specific phenotypes, like how it spreads among the population, how it causes disease, etc. Importantly, we had many different types of participants. These participants included graduate students, undergraduate students, faculty, and clinicians. And in many ways, what we have created is an experience that combines multiple elements and translates into more than just a certificate that you would get from another place like Coursera or edX, or maybe even your university, because it is a certificate that is based on practical hands-on experience. And you take away from this experience, not just the educational understanding, but a practical resource and a community that you can continue and grow with even after the program is over. So at this time, I'm going to pass it back to our Nigeria team, facilitators, Adekunle, Adetayo, and Sani Hamza, to discuss what their experience was. And I'll let Bipsa introduce you to our team in Nigeria. Please go ahead, Bipsa.
Thank you, Ilya, for the overview. So joining us from Nigeria, our collaborators and facilitators, is Adekunle Adelowoye. He is an Omics Logic trainer and a faculty at Leeds City University in Nigeria. Aditayo Aborisade is a faculty at the Department of Oral Diagnostic Sciences at Bayero University in Nigeria. And Hamza Sani is a medical laboratory scientist at the University of Lagos in Nigeria. Now, I would like to invite Adekunle to speak more about the program and his experience.
Thank you very much, Bipsa. Yes, as I've been introduced, my name is Adekunle Adelowoye. I'm a faculty from Leeds City University here in Nigeria and also a mentor with Pine Biotech. I have my background in medical laboratory sciences and currently I lecture all medical scientists, nurses, and radiographers. My area of focus is molecular biology and genomics, where I'm currently completing a PhD degree. And I'm also presently exploring opportunities in bioinformatics and big data science. A career I've found very exciting and fulfilling since coming to join the Pine Biotech platform. I'm also currently part of the OmicsLogic Nigerian team as a facilitator and program director. Pine Biotech is a bioinformatics company that is working towards providing cutting-edge bioinformatics research and education in partnership with several other universities across the globe. And one of such is the prestigious Leeds City University here in Ibadan, Nigeria. As a previous participant and alumnus of the Pine Biotech program, I'm currently utilizing acquired knowledge taking courses in bioinformatics, transcriptomics, metagenomics, to study the variation in angiotensine converting enzyme 2, that is AC2, among male and female normal as well as COVID-19 subjects, infected subjects. As we all know, ACE2 receptor binding is what the SARS-CoV-2 virus utilized in entering into the cells to infect the cells. As exciting as bioinformatics is, the fact is there is a computational resource gap when it comes to successfully running bioinformatics pipelines on data sets. And first is that big data science and bioinformatics are infrastructure demanding, as there is a need for high computing power and space to be able to run these pipelines. And just to add a perspective to that, you can imagine you have a genomics data set you want to analyze, and it's about 300 gigabytes, in size. I mean, in this part of the world, even downloading this kind of data set, file would be a huge challenge, not to talk of processing and analyzing them. So this is one of the challenges we have when it comes to, bioinformatics. Computational resources gap. Now, this of course meant a huge investment, as the gadgets and resources needed are very, very expensive. There is also the inaccessibility issues, as well as the wide gap in the expertise necessary to carry on these tasks and mentoring of those coming into the field, despite the need for these key sets. And this is where I find very, very interesting and helpful the Pine Biotech's cloud-based AI-guided server for participants to utilize in their learning journey. Aside the well-structured asynchronous courses and other learning resources, learners can immediately begin to have hands-on experience on big data analysis. Here on this slide is a screenshot of my own personal project pipelines I ran using the cloud-based AI-guided T-Server platform during my learning experience. So the present COVID-19 outbreak have come not only to test the scientific knowledge and advancement of the past decades, but also it has come to show how important these scientific breakthroughs, such as the next generation sequencing and other molecular biology technologies, have become in helping to cope with the present pandemic that threatens the very core of our human existence. And so the next important component that need equal, if not much more attention now, is skilled professionals that are able to respond adequately to these opportunities and challenges using bioinformatics. I therefore look forward to having many more professionals, university faculty and researchers, as well as students, as budding scientists and professionals, to key into this opportunity that is available by participating in the forthcoming basic Omics Logic Training Part 1, as well as many other training programs that is to follow towards becoming an expert in this field. At this point, I will equally want to hand it over to my co-facilitator here in Nigeria, Dr. Adetayo Aburushade, to say some few things also. Thank you.
Hello, good evening, everybody. I'm Adetayo Aburushade, a lecturer in the Department of Diagnostic Sciences in Bioinformatics, Kano, Nigeria. And I'm also a post-fellowship senior registrar in maxillofacial pathology in Aminu Kano Teaching Hospital, Kano, in Nigeria. By background, I'm a dentist. By clinic-based study, I'm an pathologist. Because of the fact that I love to ask why. That's why I'm a pathologist. And while during the course of my study, I was also able to see the immense opportunities in data analysis and molecular diagnostics. This is what actually attracted me to bioinformatics as a field. And the fact that you can learn a lot from a patient by looking at data from the past and predict a prognosis of that same patient by the genetic signature of the patient. You can learn why they are susceptible to some diseases, why they are healed at times, and why they fall sick at times. So it's this interest in why aspect of disease that made me become a pathologist in the first place. And I found out that bioinformatics not only helps to understand why, but also the why not. Why are some people susceptible to some diseases? Why is there cancer in some diseases? Why do some people smoke and get down with lung cancer? And some other people smoke and don't have any iota of this carcinogenesis. With the initial workshops I took in bioinformatics, you first have to learn the basics of data analysis. You have to learn either Python, before you can start answering any project in bioinformatics. But one good thing about the Pine Biotech is that you don't need to spend months learning data analysis. You don't need to spend months learning R or Python. There is already a cloud server where the only thing you need to do is follow the steps and you get the same result as if you are performing by yourselves. And at times, when you are running these pipelines in some other websites, it might take you 4 or 5 days to just get one answer. But if you can look at my own project pipeline, you can see that at times I run 4 to 5 pipelines daily. And that means in a couple of days, I get to get as many results as possible. I get to finish many projects. This is the advantage of this server.pipeline. And that's why we feel that for beginners, for undergraduates, for learners, for people that are just starting in bioinformatics, the server, it's a good way to start to learn. And that's why we feel that in Nigeria, we need a set of academicians, researchers that can make use of this pipeline to forward and to forward research in bioinformatics and all. You also have a plethora of mentors. Initially when I started bioinformatics, I could not find mentors to guide me in the path. I could not find people I could talk to about a research project or research item. But in Turbo Server, in Pine Biotech, that's not the case. You have mentors both locally and internationally, so that for each research project, you have assistants. For each research tool, for each problem, for each obstacle, you have mentors to guide you. And that's why we are glad that we can actually form a community in Nigeria of like-minded bioinformaticians or people that have interest and passion in bioinformaticians and genomic medicine to be able to create and fortify molecular diagnostics laboratory and research. Thank you very much.
Thank you so much, Aditayo and Adikunle, for your insight. So the graduates from our previous programs in Africa have not only gone through this hands-on training that Dr. Aditayo just spoke about, but also applied their learning to their own research projects. For example, Dr. Ayorinde here, who is from the University of Ibadan, proposed this project on differential transcriptome expression of malaria and semi-immune adults to a Plasmodium vivax sporozoite challenge. In a similar way, we had Niambo, who also proposed a research project on in-depth transcriptomic analysis to determine the response to inhibition of pKNA and pKNB by M. tuberculosis. So this entire training program is designed to make sure that you can first learn and then apply the logic to your own independent research projects. Here are more examples from participants of our previous programs who have also given their feedback. For example, Kingstone was part of 2 of our previous OmicsLogic programs in Africa last year. And he shares that he had a great experience with the program that we conducted. So the aim of the OmicsLogic program and the training is to help you towards your goal of working on your own independent research project. And we have some example projects on the side with which you first learn, and then you can also propose your own proposal, and we will offer you the mentor support that Dr. Aditayo pointed out. So we are going to be starting from these existing resources. And in a minute, I will show you the examples on the site. So I'm sure that by now, everybody has already signed up to the server and to the EduT Bioinfo portal. If you have not already, we will also be emailing you again all of these details, and you can then sign up at your own pace. So how to register for the upcoming program so that you can get access to this training, the resources, and the mentor support. So I will now take you through the different steps. So we have a portal, or what we call a landing page, and I'm sure that many of you must have already signed up here as you are joining today's webinar session. So this is the link again. Once you register on this link, you would receive an email from our team in Africa, and the email address would be africa@pine.bio. So let me again post that email ID in the chat, africa@pine.bio, for any questions related to this upcoming program. And as you see here, you have a form here. So if you have not already signed up, please sign up via this link. And once you're signed in, you are registered, you will receive all the details for payment via email. And once you're already signed in, you will have access to this first resource. So as we already showed you that you will get access to EduT Bioinfo, which is a first platform, right? So if you come under events, you will have an event where you can see the Omics Logic Africa program. So I'll just give it a minute to load back. Okay, so now you have the Nigeria training basic. So if you're already logged into the server, you can go ahead and check out this page. It will lead you to this page right here. I am not sharing the link again in the chat because this is the link you get access to once you're already registered for the program. So once you're here, you can see there are several sections right here. First is the forum. So the forum is where we share different communication links with our participants. Moving ahead, we have the courses right here. And we also have several different projects. So the first step to sign up for this is to register here. Now, I would like to invite Dr. Mohit to say a few more words. Dr. Mohit, if you can go ahead and talk some more. Thank you.
Thank you, Bipsha. Okay, let me share my screen. All right. Hello, everyone. A very good evening. Today, we discuss the introductory training that is designed to infer biological insights from big data and gain in-depth knowledge from guided workflows that you saw, courses, and publication-based project examples. So Bipsha also mentioned that how we can translate from this training to working and doing a project. So I would like to share a few slides about that. So to broadly describe the application of NGS data, these methods are commonly used in variety of biomedical research that you heard about today, and also clinical projects where large data are beginning to be more and more available. So one of the objectives of the OmicsLogic program is to be able to develop an independent project or gain experience from working on a project with a research team. So the next step after learning about the technology is to apply those skills to a real research project. So we follow a particular curriculum, and we talked about the OmicsLogic program, and we learned about how this is actually working for our participants all around the globe. So to describe you this in a nutshell, what we will try to learn in this entire experience is to understand how modern biology is shaped by data and these analytical thinking to develop that, and also to learn from research project examples, applying advanced bioinformatics to real problems, to gain a broad understanding of how high throughput technology and omics studies work, and also to gain solid grounds in omics processing, analysis, and machine learning. And in the end, when you have all this information and all these skill sets and understanding of the technology, it is to connect those dots, so to explore system biology through multi-omics integration. As I was telling you that the application is pretty important to complete this training, because you start from the understanding of these technology, and then you want to do work on your own research idea. So that idea could be about oncology, could be about virology, could be about neuroscience or biotechnology, where you also saw the application in the microbial world. And also again, because we are using these data science and machine learning techniques, so that those are the research areas that we are focusing on. And as you saw, that these are the people whom we have worked with, whom we have worked as a research fellowship program, and they came up with by collaborating amongst themselves, came up with these research ideas, and they were able to work together as a group and come up with a project that has objectives, results, and conclusions. So one of the benefits of doing this, and like you saw several of these examples, is to be able to complete these projects as a part of research and learn by later presenting this in prestigious conferences, leading to poster presentations, conferences. And to start this journey, I mean, you start with the basic understanding, and then you move towards application, and then publication, and so on. So this is so that the entire thing could be showcased in your profile. You can put it as in your CV, and connect with people whom you have this research interest using this community. So thank you for this for the time, and